In this video, image-based high-content screening is performed to assess small molecule-based modulation of the fibroblast growth factor Ras/MAP kinase pathway in zebrafish embryos. Transgenic male and wild-type female zebrafish are mated in a breeding cage. Fertilized eggs fall to the bottom and are collected and transferred to a petri dish. The embryos are incubated overnight, then placed into individual wells of a 96-well round-bottom plate. The small molecule BCI is added to hyperactivate FGF signaling, and the plate is incubated for 6 hours. Images from each well are captured with a confocal plate reader and analyzed using a self-designed algorithm which reports fluorescence intensities in specific embryonic structures.
Hi, I'm Hiba Kador from the laboratory of Dr. Michael Cheng at the Department of Microbiology and Molecular Genetics at the University of Pittsburgh.
Hi, I'm Andreas Vogt from the University of Pittsburgh Drug Discovery Institute.
Today we will show you the procedure for breeding, selecting, and arraying zebrafish embryos for chemical treatment in a 96-well plate. We will use transgenic embryos that report on fibroblast growth factor activities and treated with a molecule that hyperactivate this pathway.
We will show you how we use high-content screening methodology to automatically acquire, analyze, and quantify fibroblast growth factor activity in transgenic zebrafish embryos. This procedure will highlight platform technology to utilize transgenic zebrafish in high-throughput chemical screens.
So let's get started.
Two days prior to treatment, identify homozygous male transgenic zebrafish and place them into individual mating tanks. Add separators to the tanks. Then, add a wild-type female fish to each tank and leave the fish in the mating cages overnight at 28 degrees Celsius. The next morning, remove the separator. After 1 hour, move the fish to a new tank and pour the embryos into a plastic sieve. Transfer the embryos to 100 mm dishes with E3 solution and incubate at 28 degrees Celsius for 6 to 8 hours. Following the incubation, examine the embryos under a stereo microscope. Embryos suitable for experiments will be undergoing gastrulation at this time. Remove any unfertilized embryos, which will remain at the single-cell stage. Also remove non-viable embryos, which appear opaque or malformed. Then add fresh E3 solution and incubate overnight at 28 degrees Celsius. The next day, place the embryos under a fluorescent stereo microscope to sort the transgenic embryos for similar stage and uniform GFP expression. 24 HPF embryos show a visible mid-hindbrain boundary with intense GFP expression and well-formed eyes with GFP-positive dorsal retinas. These embryos are active and will move and wiggle in their chorion. Using a plastic Pasteur pipette, transfer the 24 HPF embryos one at a time to individual wells of a 96-well plate. Using a micropipette, remove any excess E3 solution from the wells. Then, using a multi-channel pipetter, add 200 microliters of E3 solution to each well. Next, add 2 microliters of DMSO to all the wells in columns 1 and 12. Then, add 2 microliters of BCL prepared in DMSO as follows: 1 micromolar to columns 2 and 7, 5 micromolar to 3 and 8, 10 micromolar to 4 and 9, 20 micromolar to columns 5 and 10, and finally 25 micromolar to columns 6 and 11. Now, cover the plate in aluminum foil and incubate it for 6 hours at 28 degrees Celsius. Add 10 microliters of tricaine to each well to anesthetize the 30 HPF embryos. Then, load the plate into Molecular Devices Image Express Ultra. Open Meta Express software. Choose a configuration with a 4x objective and maximum pinhole diameter. Configure the laser autofocus to detect the plate bottom, and use a predetermined optimal Z offset to detect the regions of interest. Set the scan area as a single site of 2000 by 2000 pixels with no binning. Select the appropriate plate type and the 488 nanometer argon laser at excitation over emission wavelengths of 488 over 525 nanometers. Then, select a well containing a vehicle-treated embryo and click Find Sample. The instrument will perform an autofocus scan and acquire a single image. To confirm proper image acquisition, randomly select a vehicle-treated well and acquire an image. If the image is in focus and shows GFP expression in the head, position the cursor over the brightest areas and verify that the fluorescence intensity is within the range of the 16-bit camera, less than 65,000 units. Repeat these steps with another vehicle control and a few compound-treated wells. If necessary, adjust laser power and gain such that brightest regions in the positive control images do not saturate. Next, in the Plate Acquisition and Control window, choose Acquire Plate to start plate scan. The instrument will automatically acquire one image per well for the entire microplate and archive them in a SQL Server database. Once the scan has finished, upload the images into Definion's developer by opening Developer with the Salinger option enabled. Create a new workspace. Then, from the File menu, select Customized Import. Next, navigate to the folder containing the images and open a sample file. Choose an import filter that recognizes image format and file structure generated by the Molecular Devices Image Express Ultra platform. Now, to process the images, open a control image and load the desired Cognition Network Technology, or CNT algorithm, or rule set. The custom algorithm used here is designed to automatically identify embryo, yolk, and head, and to quantify the brightness and area of GFP-expressing head structures. Execute the rule set to the stage of head and yolk detection to ensure proper assignment of yolk sac and head. Modify the rule set to adjust the yolk detection threshold if the yolk is too large or too small. Next, execute the rule set to the stage of bright head structures detection to ensure proper assignment of GFP-expressing areas in the head. Modify the rule set to adjust the head structures detection threshold as multiples of mean yolk intensity until the detection of bright head structures matches visual observation. Next, execute the entire rule set manually on several randomly chosen images generated from vehicle-treated embryos and positive control images from BCI-treated embryos. To run the algorithm on all plate images, select the plates to analyze and choose Analyze. Then navigate to the optimized rule set and analyze all wells using the integrated job scheduler. Empty wells or ambiguous images will automatically be excluded from the analysis. During the scan, a continuously updating heat map is shown. Both the data and images are accessible during the run for visual inspection. The numerical data and processed images are automatically saved. After all of the images have been analyzed, the data can be exported to third-party software, such as Excel, GraphPad, or Prism. This figure shows archived scan images from a vehicle-treated and a BCI-treated well with and without CNT analysis applied. Green fluorescent protein, or GFP, is detected in the eye, mid-hindbrain boundary, and trigeminal ganglia of transgenic embryos. Note that both GFP intensity and the expression domains are expanded in BCI-treated embryos. Here, a dose-dependent effect of BCI on GFP reporter gene expression in transgenic embryos is seen. The concentration required to elicit a half maximal response, or EC50, was approximately 12 micromolar.
We've just showed you how to breed, collect, and array transgenic zebrafish embryos into 96 well plates. We have added increasing concentration of BCI, a known activator of the signaling pathway.
We have also shown how to automate the imaging of transgenic embryos and have analyzed the images with a custom-designed algorithm based on cognition network technology. With this approach, we were able to measure quantitatively, in a graded fashion, the effects of a small molecule activator of fibroblast growth factor signaling.
When doing this procedure, it's important to remember to carefully choose embryos to minimize variability between specimens.
That's it. Thanks for watching, and good luck with your experiments.